Welcome to our presentation about metagenomics. This presentation will go in depth about metagenomics so you can get a good understanding about this amazing topic.
First, we will talk about what metagenomics is.
Metagenomics is the study of unculturable microorganisms in the microbial world. In metagenomics, DNA samples are taken from organisms in their natural environments because they cannot be grown in lab conditions, which means they cannot be cultured. This is due to the difference in the environment in the lab and where they grow naturally, and that most microorganisms are not independent. Microorganisms are generally mutualistic, meaning they need one another to grow. The word meta in metagenomics means transcendence, which is the focus of genes in that community of microorganisms. The word genomics in metagenomics means hereditary material, which refers to the traits that will be brought or carried onto future generations.
Our next question is, why do we use metagenomics?
We use metagenomics to gain a better understanding about simple and complex environmental samples. Since only 1% of bacteria can be grown or cultured, it is very important that we use metagenomics to learn about different organisms.
Next, we will talk about the difference between metagenomics and genomics.
Genomics is the study of one type of species, while metagenomics is the study of a whole community of species. In metagenomics, scientists look at many different types of organisms and how they are related, not just an abundance of one type of organism like in genomics.
Next, we will explain some problems and solutions in metagenomics.
Some problems in metagenomics are that originally we thought every organism could be grown in a lab, although with more research, they realized this was not the case. After many tests, scientists found that organisms are mutualistic and need to be grown in their environment. Scaling data can also be an issue since scientists work with such large numbers of DNA in microorganisms. False information is also possible. Genes could be called into the wrong strand family or even completely missed.
Our next question is, why are metagenomics important?
Metagenomics are important because in the future we will have to use this method to figure out what type of disease patients have and how to help treat or cure them. This process works in determining the type of bacteria is in the patient. It would help with bacterial disease as well as infected wounds. Metagenomics also helps us understand the communities in different environments and how they are related in the collection of environmental samples.
How can metagenomics contribute to advances in many fields?
In earth sciences, scientists use the development of genome-based microbial ecosystem models to describe and predict global environmental processes, change, and sustainability. In bioenergy, the development of microbial systems and processes can lead us to new bioenergy resources that are more economical, environmentally sustainable, and less vulnerable to disruption by world politics.
In agriculture, metagenomics improves more effective and comprehensive methods for early detection of threats to food production, like crop and animal diseases, and food safety, like monitoring and early detection of dangerous microbial contaminants. It also maximizes the beneficial attributes of microbial communities practices in and around domestic plants and animals.